From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on December 5th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 118th episode of Matters Microbial. Thanks for every view or listen, and I do appreciate every listener and viewer. Please spread the good microbial word if you would. I recently returned from the American Society for Microbiology's Conference for Undergraduate Educators in San Antonio, Texas. As you can see in this photo, I did my best to show that I am indeed proud to be a microbiologist. My colleague and friend Dave Westenberg, former podcast guest, gave me this wonderful lapel pin celebrating Angelina Hesse, who first used agar to solidify nutrient media in petri dishes. I'll put some links in the show notes about Angelina, also known as Fanny Hesse. I ran into several fellow microbial enthusiasts, including former podcast guest Rachel Poretsky, as seen here. It was just great to chat about microbiology and teaching with attendees. We're in the final stretch of the last microbiology course I'll be teaching to undergraduates here at the University of Puget Sound. So I spend time sharing microbial joy and important microbial concepts with my Micronauts. They are truly wonderful people, and I'll miss working with them. For example, here is some Lux art celebrating this podcast using different exposure times turned into a short movie. Art is by the great Dr. Jennifer Quinn. I offered to 3D print glow-in-the-dark bacteriophage earrings so students could remember how important bacterial viruses are, seen here. Conceptually, I have long been tinkering with quorum sensing experiments and demonstrations for Micronauts. That is, showing how some bacteria speak a chemical language to which other bacteria can respond. For example, Chromobacterium makes a dark purple pigment called violacine. It only does this when there are many Chromobacterium cells present and communicating. I have a mutant of Chromobacterium, CVO26, that cannot speak the chemical language, but can respond to it. So seen here, CVO26 in the center will make the purple pigment only when it is near the wild-type strain making the pigment. Please notice that the CVO26 is responding to something diffusing from the wild-type strain. It even looks circular. And it turns out that CVO26 can respond to chemical signals, language, that are made by some bacteria that do not make violacine. So maybe they have a common word in their language. Seen in this image, the Erwinia bacteria that do not make the purple pigment at all, but the CVO26 above it responds to the Erwinia signal by making the purple pigment. So yes, microbes can communicate via diverse chemical languages. I'll put links in the show notes about this central concept in microbiology. Another central concept relevant to today is that of biofilms, groups of microbes clinging to surfaces. Now, this has many advantages to the microbes making such a biofilm. After all, microbes don't usually exist in pure culture, they don't usually form single colonies on plates, and they usually don't grow in tubes of nutrient medium. Even in flask culture with shaking, biofilms are often observed, such as this flask culture of Chromobacterium. Because of the pigment, the biofilm is clearly evident. I even developed a laboratory exercise involving biofilms and Pseudomonas putida using a 24-well plate seen here. If a biofilm is made, it clings to the wells. The adherent biofilm stains with crystal violet dye, and the amount of dye is proportional to the amount of biofilm. Thus, it's possible to have a negative control, as you can see, no bacteria, a positive control with bacteria, and 2 concentrations of a possible anti-biofilm compound. Because there are 6 wells for each condition, decent statistics can be employed, and I used the systems for many years here at the University of Puget Sound to not only teach first-year students about biofilms, but also how to collect and analyze data. And I guess I talked about biofilms a bit too much. Many years ago, the Biology Honor Society at the University of Puget Sound would gently prank faculty members every year. That year, they turned my lab door into a biofilm, seen here. Biofilms are an important part of microbiology and are seemingly everywhere in nature, doing so many things. So I am extremely happy to introduce Dr. Matthew Fields, Professor of Microbiology and Cell Biology, as well as the Director of the Center for Biofilm Engineering at Montana State University, to the podcast to help us appreciate the depth and breadth of biofilms on our planet. Welcome to the Quality Quorum today, Matthew. It's an honor to meet you.
Good morning, Mark. It's great to be here.
I am so happy. What's the weather like in Bozeman?
I had to get up a little early this morning and clear some snow out of my driveway, so that's par for the course this time of year.
I've heard so many stories, and I went to Bozeman about a year ago, and I had a lovely time at the Institute for Thermal Biology.
Yes, that's another great institute here at the university.
So would you mind telling listeners and viewers what the Center for Biofilm Engineering is and what the goals of the center are?
So the CBE, the Center for Biofilm Engineering, is actually celebrating its 35th year this year. So it's our 35th anniversary. So this is great timing, Mark. And the center was originally funded out of the Engineering Research Center program out of the National Science Foundation. And that was way back in 1990. And the center is today one of the longest running centers out of that program to date. And that program still runs at the National Science Foundation. And so back in 1990, the center and the folks that were here came together and created a transdisciplinary center focused on biofilm research and everything, like you were saying, that biofilms can be and where they find them. And both not just in university federally funded research, but in a strong relationship with industry, because these biofilms can really impact industrial processes and everything that industry is doing as well. And so for the last 35 years, the main goal or mission of the center has been really to advance the frontiers of health, of environment, of energy, and at industry, that where biofilm impacts that. And I would argue that certainly microbiology and microbes, they touch everything, they're everywhere, and that a majority of the microbes that we find on Earth at some time in their life cycle, if not most of the time, they are residing in a biofilm. And so biofilms really do have implications or have impact on all of the things that we face as or challenges for society today, thinking about human health, environmental health, clean water, secure food, and energy. And biofilms really do touch all of those things. And so at the center, we have a really broad perspective on all of the different aspects that we do and what we research. And when I say transdisciplinary, we are truly transdisciplinary. And this really started from all the way in the beginning in 1990. I mean, we have affiliated faculty that are crossing through 3 colleges, almost 15 academic departments, because biofilms are these complex heterogeneous systems. Even though we always try to think of those microbes, right, or bacteria as these single-celled organisms, we know they're not. We know they're complex. They're not so simple, and they get even more complex when they start to form these biofilm structures that you can actually see with your eye. They become macroscopic in a sense, and so it takes a lot of expertise to cover these. And not only are our faculty coming across all of those different departments, but our graduate students and our undergrads. So we have graduate students and undergrads, almost 150 doing research in our labs, and they're coming from almost 15 different academic departments, and they're all working together. And that was one of the things, I've been at the center for 18 years, and it was truly one of the things that attracted me to come to a place like that not only was doing microbiology, but was doing microbiology in the context of all these other disciplines and questions. And that's what I love about being here.
I have to tell you, it's historically known that when you bring people from outside of a discipline into that discipline, there are often a real blossoming of interesting ideas. Look at the very history of molecular biology, which was essentially founded by quantum physicists that moved in.
Absolutely.
And that is no different here. I have been in love with biofilms since the 1980s, to be honest with you.
Yes.
And it's funny. You heard a little bit about the simple biofilm assays that I would do with first-year students, and I would be biting my microbial tongue because it's not ever going to be just one species.
No.
And I remember when people were first studying this, and I would say, but that's not how biofilms truly exist. And they'd say, well, I'm looking at 3, and I would kind of not say anything. And that was the first time after that I met Bill Costerton, who we both knew a little bit. And he really turned my head around on this subject about understanding them as being communities. And we've seen previous podcast guests talk about that too, these emergent properties of bringing different microbes together. It's not just a surface. It's easy to call them. And I believe Bill was called the king of slime at one point.
Yes.
And that's not what it's about at all. No. And so when I show you Chromobacterium clinging to the side, and by the way, it doesn't do that if it's not making the pigment, which I found interesting.
Interesting.
Yeah. So what I'm getting at is that it's not as simple as what you see, say, in a running stream. And my big fear is that people are just going to focus on biofilms that cause disease, which is a big deal. But as you allude, it's everywhere.
It is everywhere. And we run into that too. And certainly a lot of the companies we work with and the work that we do is about microbial control. And if you want to control microbes, a lot of the times it's going to be as a biofilm, or biofilm as a part of that issue. But we also try to think about biofilm. Okay, microbes are here. They're very hard to control. You know this, and we every microbiologist know this. And biofilms are going to be a part of the system. How can we turn that on its head and actually use that to our advantage? And can we begin to understand enough to actually have positive or probiotic biofilm? Where that's in its infancy, and we're a ways from that, but certainly folks are thinking about that. And can we, you've heard, at least this came up in some of our conversations about engineering biofilms and engineering microbial communities. And can we begin to know enough where we can have the right microbes, right, to keep out the other problematic microbes? And this is what's happening even when you think about the human gut, right? And a lot of people, and I know a lot of students, your students are probably the same, they're very interested in this gut microbiome, how it impacts human health. You're really just thinking about how microbes impact their local and regional environment. And we now, the host, the human, is the environment. And there's a lot of things going on there that we are just starting to scrape the surface on. But that's routinely, right? We know that gut microbiome is doing a lot of things. And part of what they're doing is occupying that niche space, not only doing metabolic exchanges and such, but also preventing then some of the other opportunistic pathogens or other things that we don't want, and excluding them through exclusion and niche exclusion to not allow them to grow. And we're just starting to scratch the surface on that. And could we start to bring that concept into other environments?
It's interesting you'd bring this up. And we could talk for hours on this, so I'm really watching myself. I do a simple exercise with students where we try to find out what's living on the walls of their reusable water bottles.
Oh.
No, it's and I wish I had the funding to be able to really look at how those communities change, because they are communities. And I talk about oligotrophy, and I talk about biofilms, and I find antagonists from time to time. I'll find microbes there, bacteria, that make antimicrobial compounds. So that's the important thing, I think, for people to understand about biofilms. They're a dynamic assembly, and they're an ecosystem.
They are, all in of themselves.
And I would even argue that kefir grains are biofilms.
Oh, yeah, yes.
Kombucha is a biofilm. I
Yes.
mean, it's everywhere.
No.
So if you don't mind my saying, I believe you have a slide, because I've used it in teaching, that shows the biofilm occurring in different things, including medical and going to industrial.
Yes. And so we try to, we have lots of these different types of images, and I'll try to bring this into the conversation here at some point. Imaging is really important here and we work really hard here at the center to maintain a state-of-the-art imaging facility. And part of that is to bring home this idea of the beauty, right, of biofilm and what it is and where they're at. And so, yes, I use slides when I talk that show this wide breadth of anything from actually a water line from a dentist office, and you open and you split that tube open and you look at the biofilm. This would be at any, not to speak, this would happen at any place you would go, right, where there's a water distribution line. Biofilms are gonna be a part of that. That's in the premise plumbing that's in our houses right now. And it goes certainly to other industrial processes. Anywhere where you've got an interface, be it a solid or a liquid, a solid and a gas and a solid. And it's not sterile, which is about everywhere. Microbes are going to go and they're going to collect at that surface. A lot of the times driven by just the physical fluid dynamics of the way that the system operates. This is what cells are going to get driven to that interface. And that a lot of the time is where nutrients are. And probably how originally biofilms were selected to even become biofilm. If we think about some of the earlier, in early life and when we first had microbial cells, we have fossil evidence that if you think about a stromatolite, that's a biofilm. And so, biofilms were in existence for almost as long as we think microbes have been here. And so it happened early, in evolutionary history. And so anywhere you go where you've got those interfaces, which is about anywhere, again, from a stream bed, and you got rocks, and you mentioned the stream slime, that's a biofilm. The film that you feel on your teeth after you haven't, you've gone a little while before brushing your teeth in the oral cavity, that's a biofilm. A medical device or a prosthetic device that goes into the human body, be it indwelling or even say like an indwelling catheter, those are biofilms that form. And so they really do go to that. I think. So I've seen a statistic in a piece of literature that I could share with you, and they did some back of the envelope calculations. We think that let's do a microbial cell count for Earth, right? And we've all seen this number, right? It's anywhere from 10 to the 29th to 10 to the 30th, a total cell count for Earth. If you do a back of the envelope calculation, plus or minus a few cells, Mark, right? And then if you go and look, well, where are the majority of those cells? And how prevalent are biofilm in those environments? The calculation comes to maybe 40 to 80% of microbial cells on Earth are residing in a biofilm.
Yeah.
And so this is a very dominant way of life for these microorganisms, and they've been evolving this way for a long time. And as you were alluding to, quite complex, the communication. Really, to me, that is where biological communication and cooperation was being forged. Because it happened so early and they were probably so diverse. They were figuring out how to work together. And this was really crucial to biology. Now, that's a hard experiment to go back and prove, right? That's really, really, really hard to do, if not impossible. But to me, at least from my biased perspective and where I am and the work that I do, I think biofilm certainly had a role to play in the further, as biology was evolving and coming up. What is, when you think about a biofilm coming together in these microbial cells, and it's where nutrients are concentrated. They do get some protection from changing environment, in salt. We think about photoradiation, or we think about oxidizing agents or reducing agents, or pH changes or salt changes or osmotic stress, desiccation. We think about all of these things that biological systems are dealing with. biofilm began, was partially an answer to dealing with those environmental stresses. And so I think biology was forging a way to deal with that. But when it did that, it also was starting to cut itself off. And so you start to now have diffusion kinetics coming into play, right? And so now I've got to start bringing things in, and there are cells that are deeper down into this biofilm that I've developed, but I also got to get waste out. And so if we think about like what the human body does, we have this complex circulatory system, and I can have gas exchange and liquid exchange, and I can move it internally within this biomaterial. To me, biofilm was starting to figure out those problems. Now, does biofilm have a circulatory system? No, but it is starting to try to modulate this idea of reaction kinetics versus diffusion kinetics and ways that cells were trying to adapt for that. And so when I think about, again, like the crucial aspect of what biofilm represents, it was the beginnings of all that. And so it tells us something about how does biology respond to changing environments, not just on a short time scale, but very long time scales.
It's interesting you would bring this up because we had a guest of the podcast a few episodes ago, Joshua Shrout from Notre Dame. And he had some beautiful images of mixed biofilms of Pseudomonas, and it might be E. coli. I'm sorry, I don't remember off the top of my head. But the point is that they're battling for space, and you can see that happening. And there's a point of view that suggests that things living together are in harmony, and there's a point of view that says that things that are living together are battling one another. And when students ask me about this kind of question, which do I think is correct, I usually say yes, because I think it is contextual. For listeners and viewers, there used to be this thing. This is how one of the first times I met my now wife, she had one of those big crocks and a giant bottle of water in it, and would like get the water out of it. And I was going to show how strong I was to impress her by changing that. And so I got the big bottle off, and I started cleaning the inside of the crock. And my now wife says, "Why are you doing that? It's just water." And I took my finger and went, and it was like brown.
Yep.
And I actually took those samples. They're mostly colobacters, which are known to live in those kinds of low nutrient environments, presumably fed by dust, right?
Yeah.
And so they're everywhere, and you cannot get away with it. They were here first evolved, last extinct.
Yeah.
And I just wanted to say, as you talk, I wish that I had been at an institution with a lot of seminars, because I think giving a whole course on biofilms, as we'll see today, would be great.
Yes. No, they really do touch a lot of things, touch a lot of different environments. And I think that was what your original question was, right? About all of the places that we go, we see biofilm, and it's from inside the human body to outside the human body and everything else that we interact with.
Agreed. Yeah. It's funny because I think we should move to the second bit. You have a lovely image of how biofilms develop. Would you like to walk us through that?
Yeah, and there's actually over the last couple of years there's been some revision of that, right? Which we always do in science, and that's the way that it goes. And so one of the images that I shared was what was referred to as the original 5-step model for biofilm development. And this idea that you had planktonic single cells, or even a small number of single cells, they find a surface or get, whether that's directly or indirectly looking for that surface. Once they have ways of sensing, right, that they really actually have, and we're just, there's active research in this area, and we're still a lot to learn about how a microbial cell senses that now it's at a surface, right? It feels physical force. And we know in biology there are mechanosensitive proteins that actually start to allow cells to feel that their environment has changed in that way. And so they sense that they've interacted with the surface and or each other, and they begin to then change their physiology and their phenotype. They begin to down express genes and up express genes that allow them now to stick to each other and to that surface. And then one of the hallmarks then of biofilm is this matrix material, or this exopolymer that we know now from a lot of different organisms is not just carbohydrate that forms these polysaccharides, but it's also proteins, it's also nucleic acids. And this matrix then starts to hold the cells at that surface and actually can resist um, the flow field that typically we're thinking about these as a liquid-solid interface, and there's some type of a flow, and there's a force to that. And this matrix material has then viscoelastic properties that resist that flow and actually help keep the cells there. And then that biofilm will spread along the surface and get bigger, anywhere from, it could be a monolayer to a couple cells deep to hundreds of microns deep. We know that, and you get these biofilm structures that are typical. Now, to really be truthful, we are still in our infancy of biofilm understanding, because a lot of what we think we know about biofilm is really on a couple microorganisms. A lot of it's Pseudomonas, and you mentioned Pseudomonas earlier in the beginning of the podcast and what you do with your students. A lot of work has been done with Pseudomonas, and rightly so. Pseudomonas is a widely distributed organism, important in almost most environments that we look, both the human body and outward. And so some Pseudomonads will make these mushroom structures, and so they actually have a stalk and they'll balloon up. Sometimes they're just monolayers, sometimes they're just these valleys and peaks of just an undulating surface. But at some point, whatever that biofilm looks like, it will then get big enough, and then in that environment, then there is some shear force, and the cells start to break off. And this could be as aggregates or single cells, and then those cells are then going downstream to whatever environment they are in. They might live as planktonic single cells again, they might live as an aggregate, they might land at another surface and start the whole process over again. So then if another slide that I provided then is actually from a paper a review article that was published in 2022, Sauer et al. This is Karen Sauer at Binghamton University. And in that, she worked with colleagues here at the center, along with some other colleagues in the biofilm field, and they sort of revisited this biofilm model. A lot of it is still the same. You can get these single cells, they go to a surface, the biofilm grows, it comes under force, it breaks apart, and that kind of cycles. But now there are these ideas of aggregates, and we can go to a lot of environments, and sometimes the aggregate might be the only biofilm that's there. And we see this in water bodies, we see this in freshwater or marine. You see it in cystic fibrosis patients, right?
Yes.
And the aggregates that form from the bacterial infection. And so those aggregates themselves are the biofilm. They could go to a surface and start another whole biofilm process, or they could just stay suspended in an aqueous state, in an aqueous phase, and just exist as that aggregate biofilm. And so what this revision of the biofilm model came to be was just working this idea of aggregates into the biofilm life cycle. And that really came out, I think it was actually before COVID, it was pre-COVID in 2019, there was a biofilm bash. And it was a group of a bunch of people from the United States as well as from Europe and other nations that came together. It was probably 50 to 60 people, and we all went to a retreat and asked. We had like our own little workshop and asking about where's the field, what do we need to do to move the field forward. And one of these notions that came out of that biofilm bash was working aggregates. Now we had started to see and realize that these aggregates were an important part of the biofilm life cycle, and that's where this notion of revising the biofilm life cycle came from.
This kind of is so interesting to me because I start to think about the evolution of multicellularity. And I am not trying to overstate things, which I know a lot of scientists do because they get excitable. But the idea that it's not just one cell that is causing this, but groups of cells together, that's how we happened, essentially.
No, I agree. And when I was talking a few minutes ago, I was sort of alluding to that. And I think you're right. We do have to be careful because we can't really do the experiment. They're hard to do. But I can't help but at least think and at least ponder that this notion of, there's certainly examples of not just let's take this one step back and think in early evolution, we got to a point where there were bacteria and archaea.
Right.
And we're even at a point now where there certainly is data to argue that we may really be simply in a two-domain
Mm-hmm.
life. That
Mm-hmm.
eukarya could be seen as a sister lineage within archaea, which is quite fascinating and really turns things on its head a little bit. But let's say we got bacteria and archaea, and whether eukarya is a separate line of descent or within the archaea, we know that bacteria and archaea come together in biofilm. And so once you put these cell types, biofilm was the place it could have happened, right? They're in close proximity. They know each other's there. They're figuring out how to, like you said, sometimes they're cooperating, sometimes they're competing. How does biology manage that? That was probably being laid out in biofilm. And it's really hard to argue how that couldn't be. I have no idea if that's true or not. And again, those are really hard experiments to do, but I can't help believe that it didn't contribute to the process somewhere.
If you think about it, we're really just talking about ecology. And I say this as a non-ecologist. I want to be very clear on that. But how do you study ecology? And it's such a complicated system, has so many different moving parts, but then again, so do biofilms.
Oh, yes.
So we already know from Rich Lenski's work and colleagues that it's possible to study the process of evolution using bacteria. Well, we might be able to make real progress in larger ecological questions by studying something that's manipulable in some sense, although we can only have a certain number of members that we manipulate.
Yeah. Now, this reminds me, and certainly some folks have started to bring the concept of biofilm into these evolution experiments. And there have been some very interesting papers out there using biofilm reactors. Any reactor is challenged to run, you're aware of, right? To maintain the system for a long period of time can be very challenging. And so to do long-term evolution experiments, thus, are challenging. Now, that makes me think, though, of and I just crossed paths with him at ASM Biofilms a couple weeks ago, up in your neck of the woods, up in Portland.
That's right.
And Vaughn Cooper has done some interesting experiments. And I don't know, have you had Vaughn on the show?
We have in the first year, and his evolving stem that he does for is just wonderful.
Yeah. And so I was just that whole concept, right, of just simply using a glass bead and just moving it every 24 hours to a new tube of media. That glass bead allows the biofilm to form, and he can just simply, and that's that's way simpler than reactor systems, and yet simple and elegant.
So I want to throw something at you really quick, and it is related. Roberto Coulter and some other folks at Harvard not long ago, but a while ago, were able to show that organisms isolated from nature would give different colonial phenotypes than those that had been domesticated in the lab. This makes me think, for example, the undomesticated colonies are all crenated and look all complicated versus very simple and smooth. And now I'm wondering, is the same thing true for biofilms? Because I've always wondered about colony morphology, because how often do you see colonies? But you do see biofilms everywhere, and I'm wondering if the biofilm architecture changes.
Well, so are you? If I'm following your track of thinking here, we as microbiologist undergrads and such, right? We have drilled into us, unfortunately, for better or for worse, like pure culture, right? And so when you ultimately get that pure culture, and then even look at that pure strain as a colony on an agar plate, right, you've sort of taken away a lot of internal diversity or inherent diversity. And yet,
yes, you have. Yeah.
And so it makes me think, I think absolutely you're right, that if we were to simply take an organism, let's say, and really we have to think about in the environment, we're seeing this in environment, and we do this work in some other environments that I work in where we're trying to understand population dynamics. Even in an environment like groundwater,
right,
an immense amount of diversity, and even within the same species, there appears to be what we would refer to as subvariant pools. There's all these slight variants, right? And so if you when we're our traditional methods have been to pick those apart, right? And that's not how they're functioning as a unit when they're out in the environment. And so I think exactly, if you were to allow those organisms to be together in a biofilm, what would that do to the biofilm?
Yeah. So in other words, if E. coli, standard E. coli MC4100 that people use in the lab does form a biofilm, because I've done it, and it has a particular morphology to it. If I go back to an ancestral form of E. coli more recently isolated and not been characterized as a lab rat, would those biofilms differ? And almost certainly, because the colony morphologies do. So I'm not trying to claim that colony morphology is the same as a biofilm, but there might be things in common between them.
Oh, I think so. And we think about a colony. A colony on a plate is a biofilm. It is growing at an interface, right? It's a
Yeah.
solid gas interface. It's just a different type of biofilm, for
Yeah.
sure.
I got carried away. I'm so sorry about that. And I want to be aware of your time. So I thought if you wanted, could you talk about some of the exciting work that you've been involved with now?
Well, gosh, that's hard because I certainly have the work that I still try to do in my lab and with my graduate students, but I also am very fortunate that I get to serve as director at the Center for Biofilm Engineering, and so I get to think about a lot of different research that folks are doing, and there's so many things. I would
So how about like something having to do with human interface versus a non-human interface?
Okay.
So maybe two different things, maybe.
So one would be, we continue to look at the center and think, and obviously there's a lot of interest in where biofilms impact human health. One of those would be chronic wounds. And so, for instance, when we think about in a diabetic ulcer, those chronic wounds, whether you're a diabetic or not, folks can get a chronic wound that's very difficult to heal. And what typically is happening is that there's a biofilm in the wound bed, and that doesn't allow the host, the human cells, to heal itself. That's impeded or drastically slowed down. And so the last time that we sort of tried to tabulate these statistics, chronic wounds are thought to impact almost 6 million people a year in the United States. A lot of these are diabetic patients, and it ends up being that when you get this infection, this biofilm is in the wound bed, not only does it impede healing, but then that biofilm itself can't be really treated with antibiotic. And so it doesn't heal. And this ultimately ends up, results in about 60,000 amputations a year, costing almost $20 billion in the United States. And so that's just one example of the impact then of a biofilm in the human environment that has dire consequences. Another example would be nosocomial infections, and these are infections that patients acquire once they come into the hospital. There's almost 2 million of these infections a year in the United States that result in approximately 90,000 deaths. And of these, implants or something that's going into the human environment account for a drastic majority of them. And a lot of the times it's an endoscope, it's a catheter, it's another type of device that goes into the human body that remains there for a while, if not permanently. And it can have dire consequences, right? Even if a couple cells are there and they can establish them, so it's an arms race. And one way that we think about even a joint implant coming into the body, if there's a couple cells there, something happened and it goes into the body, and certainly we have an immune system and macrophages that are trying to find those invaders, and if they find them and can clear them, everything is fine. But if that doesn't happen and the balance tips towards the bacteria and they start to grow big enough and form this film, they become resistant to the macrophages in the immune system. It becomes difficult for the immune system to clear. Now it's a problem. And so that would be two examples, right, that that we really worry about in the human environments. And I'll if I can, I'll throw one more in because I think
Please.
it's important. It's important, and it's one that's coming. we're well aware of antimicrobial resistance. It's an impending challenge that we have coming. The NIH report that came out 5 or 6 years ago stating, with further development of antimicrobial resistance by the year 2050, which sounds like a long time from now, but that's only 25 years away. We could be at a point where there are 10 million people dying of infectious disease in the United States because we can no longer clear those infections with an antibiotic. And so that's a big problem that we have to figure out. A lot of the time in environment, so not only, and I just gave that example, like in a catheter or on a device, we get this. Biofilms are a crucial part of that antimicrobial resistance. When organisms are growing as a biofilm, they're more resistant to these biocidal treatments or static treatments. And we know that within biofilm, and this is an active area of research, they actually can develop antimicrobial resistance.
Yep.
So it's an important part of this antimicrobial resistance challenge, and one that we all need to be keeping on the radar. And I know a lot of people are, and they're working towards that. Thinking about positive biofilm, I had one area where we've been using that's technology that's at least come out of the center, and we have researchers in civil engineering and mechanical engineering where they use microorganisms, as I think I was alluding to a little bit earlier in our conversation, that can basically biomineralize, and they can take sand and turn it into a material very similar to concrete. If you think about concrete, it's one of the most used materials on a global scale, and almost 10% of global CO2 emissions come from concrete production. And so if we could start to find alternatives using native material and turning that into a structural material that we could use, there's a big impact there. And we actually have there's a company that was spun out of the center that actually is using that technology to seal orphan gas wells, so they no longer emit their greenhouse gases. And so that's a great use, and we're trying to move towards that and find different uses for that technology. Another example that I actually am involved with and that we work with is thinking about algal biofuels. And when we know that we can grow algae, they can make lipids, and we can harvest those lipids and those oils to make biodiesel and other types of fuels, maybe even a sustainable jet or aviation fuel. We're trying to always optimize that process. We know that algae, just like us and all other organisms, have a microbiome, sometimes referred to as the phycosphere or the phycosome, and that those microbes are interacting and doing things with the algae to ultimately help make that process more efficient, fix CO2 in a light-driven process, recycle nutrients, and that microbes play a part with those algae. And so they actually are within this matrix that the algae will sometimes make to have those algae in there with them, those microorganisms. And for instance, we know a lot of green algae, they can't make B vitamins just like us. And the microbiome that are there with them, they're doing some of the same things, and they're providing other nutrients. In turn, the algae are fixing CO2, and a little bit of that carbon comes out and feeds the microbes. And so if we can get happier algae, maybe we can get them to make more lipid and biofuel, and then ultimately impacts our biofuel production. And maybe they're just more stable cultures. We can begin to then use alternative sources, right, as a carbon reserve to satisfy some of these energy needs that were coming through. And so one more example that actually got me, I was fortunate enough to work with as a postdoc when I got my postdoc started at Oak Ridge National Laboratory. And we were trying to understand the microbes in groundwater and in the subsurface environment. And if you think about an aquifer and groundwater, that groundwater is moving through a porous matrix, the sediments. There are microbes on those particles, right? They're not big, thick, slimy things that we think about in streams and creeks, but they are around those particles, and they're forming films, and they're a big part of that water quality and doing things that impact that water quality. And so I was part of a project that was actually looking at nitrate and uranium contamination. Yeah. And we were able to stimulate the microbial community that was in that subsurface to help start to treat that uranium waste. And it was one of the first times in a field demonstration where we dropped the uranium levels below drinking water level standard. And that was because we tried to understand what microbes are there, what food do they need to be fed, and how can we feed it at the right way. And so we stimulated that microbial activity and actually was able to treat some of that waste. And so that's in an area called bioremediation, and that's an active area that a lot of folks study. How do we go into different environments, understand the microorganisms that are there, work with geochemist and hydrologist and environmental engineer so you can understand water and how it moves and what's in it and how that impacts the microbes, and then in turn, how did that impact water quality?
Now, there's a lot here, and I'm really grateful for you to put this together, because one of the things that I've been most concerned about, and I want to say this very carefully to listeners and viewers, I am in no way minimizing the excellent work relating biofilms to certain diseases like cystic fibrosis.
Yes.
But you've also learned about other potential negative health effects from what we've been talking about. And then I also wanted to really, really emphasize that biofilms are ubiquitous, and there are some that have nothing to do with us at all, some of which, in fact, are very helpful to us, as we've heard. I am going to be including links to the center, and I've been carefully taking notes as we've talked, and I'll put up I'll post to various papers involved in this kind of thing. I don't know how many listeners and viewers are interested in those things, but they will be in the show notes. This is exactly what I wanted from this whole session, in fact.
Well, I'm always ready to preach the biofilm gospel.
You know, it's and I think, you know, I my goal in life is to become a microbial evangelist. So you're the biofilm evangelist in a way, right?
Yes, I feel they're very, very easy to work with, and I should say easy to talk. I should say easy to talk about. Not always easy to work with in the sense of trying to do it the right way. But we have so many excellent people and students here at the center doing great work, and I'm glad that I'm very fortunate that I get to share that.
It's a wonderful thing to think about because they're everywhere and they do so many things. Just a couple of things to occur to me as we're talking about. You find in certain caves here on the West Coast what these charmingly are called snotites, which are these hanging biofilms that are using hydrogen sulfide as a source of electrons and small amounts of oxygen. They generate sulfuric acid. So they hang from the ceiling like stalactites, but they're made of this biofilm. And there's a whole community associated with it. I'll put a link in the show notes. And they're apparently, and I love this, pH 0, which to me means your pH meter is broken. Yeah. But what it means is you can't go in there without a, hazard suit. Yes. Or you'll get physically burned by this stuff. And that's something that always grabs people's points of view. So I'll put some links in there. But in addition, and this takes me back to early days in my career, the Office of Naval Research was really interested in what they then called biofouling.
Yeah.
And then they were noticing that on boats in the water, apparently before barnacles or anything else can grow, there needs to be a biofilm of certain bacteria that in turn send out settling signals to some of the other organisms in the ocean. So studying that has definite importance in all kinds of ways.
No, and we have historically at the center done some work in that area, both on fouling of marine surfaces. Also sort of related to that even is then not just fouling or is deterioration or degradation of metal surfaces. And that's obviously then working its way into the oil and gas industry. And when you hear about a pipeline failure, that could be biofilm related. I think about half of corrosion is related or microbially induced, meaning there are microbes living on the inside of those pipelines and they're slowly eating away at the metal. And eventually the structural integrity gets to the point where it's compromised because these are under high pressure. And that's what causes these pipeline bursts. But you're right, back to your original point on fouling in the marine environment, it's a huge cost on trying to prevent the drag that's caused by these films that form, and it is a successional event. and you get these early films that form that then bring this other macrofauna that ultimately causes this drag. And for instance, even in why naval research is interested in this for a while, you know, obviously the United States military has a large fleet, and they're in the boats that they move and the consumption of fuel. I've seen statistics where it says that this drag on the boats can actually cause an increase of up to 30% fuel usage.
Yep.
And so they are definitely interested in trying to keep that drag down.
So this is just such a fascinating issue. And again, when I would teach students about using Pseudomonas putida, just because it makes these wonderful biofilms and they were easy to measure, and sometimes I would ask them the question, what would be interfering with biofilm formation? Was there a compound you think might be involved? That kind of thing. And it was fun to watch them wrestle with that and use the statistics. But it's important for people to understand that studying one type of organism forming a biofilm is not how biofilms naturally exist.
No. And that was, yes, a point you were making earlier. I would say and I alluded to this a little bit, the field has really moved over the last couple decades, and a lot of it's based on Pseudomonas, and this is how fields progress. There's been a little bit of work in strep and some work in staph because of human health implications. There's a small breadth of different organisms that have been used over the last decade or two, but there's been a big push, not just at the center, but other researchers across the United States as well as the globe, where they're trying to do these multi-domain, not just multi-species, but multi-species, multi-domain biofilms, where can we start to allow the biofilm to have the complexity that's inherent to what we think is actually in situ in the real world. It becomes difficult. We can certainly grow mixed species biofilm and mixed domain biofilm, but then just the variables, right, start to balloon. And just these little responses or tweaks that they're responding to, then you just don't get the same thing every time. And then it becomes very difficult, or I should say more challenging, to do experiments and then interpret your results. And that's why we had to start with monoculture and monospecies. But we are getting better at moving towards more representative biofilm that allows some of that complexity. And a case in point would be, over the last 4 to 5 years, we've been actually doing a project with NASA. And NASA is worried about, or what they're trying to make sure of, is that when they have water distribution lines for long-term space travel, they know they have to control microbial growth, and biofilm is a big part of that. And so at the center, we've actually been working with isolates from the International Space Station, and it's multiple bacteria and fungi. And we're actually bringing those together in what we would refer to as a synthetic community to represent what we think might really be going on in the water distributions lines, and then allow those biofilms to form, work, and then test, are there ways that we can prevent those biofilms from forming? Or if they do, then can we treat them and get rid of them?
So this is one of those areas where it's like juggling too much at once. There's so many things. I believe there's an old parable about giving a donkey too many things to eat, and it like dies of starvation because it's trying to pick which ones. Yeah. Because actually that's where we're at. There's so many interesting things about it, and you're so fortunate to be working in such a center.
Yes, no, I really do feel fortunate, and I get to interact again, as I said, working on so many great things, but with so many great people from so many different fields. And our students are wonderful, and they're here, and they're the ones that really drive us and keep us excited and bring all these different questions at these different systems.
So as we start to run toward the end of our session, there are a couple of things I like to ask guests. And the first question is, what is the path that took you to becoming a microbial scientist the way that you are?
There are probably two events, right? And these are always hard, but and I'm certainly at a point in my career where I've been doing this a little bit and thinking about my last 30 years where I've been fortunate to be a scientist and who the folks that helped me get there. I would say that when I was in middle school, And I know that I was always interested in science as a young kid, but I didn't know exactly, I didn't know what I would end up doing. But when I hindsight and I look back to being a middle school student, trying to come up with a science fair project, and I was just, and the science teacher gave me a book of different examples of things to do, and I came across this one where, and it was during the winter,
Mhm.
and I grew up in northern Kentucky, and we had winter, and so one of the experiments was to go find a pond or a lake, and it, that pond or lake looks like it's pretty static at that in the middle of a winter. There's not a lot going on, and so they said that get a jar full of water and a little bit of sediment, and then put it in a windowsill indoors, and then slowly watch it come back to life and wake
Mhm.
up. And so the teacher allowed me to do this, and we set it up in the window, and then I would come in early every morning, and she allowed me, Mrs. Gardelli, was my middle school science teacher, and she trusted me. She let me come into the classroom. There was no one there, and I would get a microscope. And every day I would monitor the things that would start to wake up in that water and all of the different microorganisms. Particularly protists. There were cysts there. They started to come up and alive and become moving around. And I sort of documented that over a month. And the notion that, it looked like just water and dirt when I
Mhm.
took it, and that this notion that when it came to life and there were all these things right under my nose that I didn't know about, really, I think, awoken me to this microbial world. And then
And it is.
yeah, no, for sure. And then, move forward now. I was an undergrad. I was a biology chemistry major. And I was actually a pre-vet student, and I thought I wanted to be a vet. And I had to take a microbiology course as a prerequisite for going into vet school. And I took microbiology, and I was in the lab, and the instructor announced that they had a job to work in the biology prep lab because there was a student that had been injured. And actually, in those days, they were still using glass Petri plates.
Yep.
And one of the undergrad students was washing a glass Petri plate,
Mhm.
and it's cracked, and it cut some ligaments in her hand. And she got it operated on and was fine. But for that reason, they were looking to hire someone new while she was out after her surgery, and that person was me. And I was like, okay, I could use a job, and I needed a little bit of money. And I got into that lab, and I was making media and learning more about the cultures and the microorganisms. And I found that it was a passion for me, and I got even more interested in microbiology. And I knew that when I graduated, I kind of forgot about vet school. Um, I really liked being in the lab, and I knew when I graduated, I said, okay, the only thing I knew for sure is I just didn't want to go out and get a real job. I kind of like the school thing, and so then I started down the path of grad school, and I went and got a master's degree, and I heard people thinking and talking about PhD, and I was like, "What's PhD?" And then they told me, and I was like, "Oh, that sounds great. I'll try that." And here I am, 30 years later.
It's really interesting because I run into people all the time that think there's only one way to become a microbial scientist or a scientist of any type. My poor parents, and again, I miss them terribly, but they were raised on farms. They had no idea about any of this stuff, but they supported me. And as you mentioned, in middle school, junior high school for me, and all of that changed my life in so
Yeah.
many ways. And it's because of those teachers, incidentally, that are tolerant with our questions
Yes.
rather than just checking off the boxes that the districts push on them. Let's be
No,
clear.
no, for sure. And it was really
Yeah.
cool where I went to undergrad at Western Kentucky University, and
Hmm.
I actually got to go back there last month, and I gave a seminar, and the guy that
Oh.
hired me in the biology prep lab was still there. And I got to go back and see him and visit with him and thank him. He took a chance on this 18-year-old kid that didn't know much, and let me come into the lab and start messing around. And it's people like that, right? When you get a chance to think back, that just gave you a chance, and you never know what might happen.
You do. You never, never know. But Pasteur said, chance favors the prepared mind.
No, for sure, for sure.
And also, when the student is ready, the master will appear. And the microbes did appear,
Yes,
right?
they're waiting just to be observed. They're always
That's
there.
correct. Well, they were there before, they'll be there after. That's the motto here, first evolved, last extinct.
Absolutely.
So the only other thing that I'd ask you, if you don't mind, is what kind of 3 takeaway lessons from this session would you tell listeners and viewers?
I think one would be, certainly microbes are everywhere. They're abundant. They're the most abundant life form on Earth. Most of that microbial biomass is probably in a biofilm, at least at some point of it in its life cycle, if not most of it. And so biofilms have been here a long time, and they're still here today. They impact a lot of things that we do, both directly to us, but everything else that we touch and we think that we care about. We certainly hear, and I think you've alluded to this, we hear a lot about the negative parts of microbes and in biofilms and what they do and how they can harm us. But there's a lot that they also do. And I always have thought about if all of a sudden, if we were to wake up one morning and microorganisms were gone, we'd have snapped a finger and they were gone, we'd be in big trouble. They've been here a long time. They impact a lot of things just on the basics or fundamentals of geochemical cycling and nutrient turnover and acquisition. And so this is a microbial world, and we need to appreciate that. And sometimes it can come back to bite us and hurt us, but we have ways of tracking it and trying to understand it so that we can control it. And then how can we also appreciate the positives that they do? And how do we turn that to our advantage and appreciate what they do and help them help us?
It's funny that you said what you just did, because every year I commission art. I'm not an artist, but I commission art for my microbiology students to have on buttons and T-shirts. And one year I was reminded of this great thing that happened in the early 1960s, because Frank Sinatra, the ancient singer and actor from a long time ago, not so long for me, had been acting up in Las Vegas. And somebody asked Dean Martin, the singer and actor, about his antics, about the antics of Frank Sinatra. And I can just see Dean Martin do this. He like takes a drag on his cigarette and he says, "Hey, it's Frank's world. We just live in it." And I had a friend create an image of Frank Sinatra made of microbes.
Oh.
And it's a microbial world. We just live in it. And and right now that's on the screen, that image, and I will send you something with that image.
Oh, that would be awesome. I'd love that. That's a great way of putting it.
Well, it really is true. And I want to thank you so much for this fabulous session. And I want to wish you and yours the very best. And I hope we run into one another soon.
Oh, Mark, and I would love to come back anytime and talk more about biofilm.
Thank you so much.
Thanks for having me.
This has been Matters Microbial, a podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as always, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Matthew Fields can be found in the Department of Microbiology and Cell Biology, as well as the wonderful Center for Biofilm Engineering at Montana State University in beautiful Bozeman, Montana. Many, many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you all next time on Matters Microbial.